presenting jointly with her PhD supervisor, Dr. Charlene Janion-Scheepers. And the title of their talk is Arthropods on the Move, Mountains as Refugia. Thanks, Lucy.
Thank you so much for the introduction. So yes, I will present my PhD study, and Dr. Charlene will talk more about the impediments of morphological identification and how DNA barcoding is very useful to us, and then more on the species cards. Okay. So I'm just going to give you a background on arthropods. So arthropods provide vital ecosystem services such as pollination, nutrient cycling, as well as seed dispersal. And they're important indicators of ecosystem changes since they're sensitive to environmental changes. So there's been a growing concern of considerable insect decline worldwide, and these declines are mainly attributed to pollution, climate change, land transformation, as well as invasive species. However, studies for Africa have mainly been focused on short-term rather than long-term studies that will provide knowledge on which global change drivers are important for the changing biodiversity and arthropods assemblages over time. So this is especially concerning as South Africa faces similar threats. So if you look at this figure, you can see that the review papers that have been published are mainly restricted to the Northern Hemisphere. So we don't really know if in the Southern Hemisphere we are facing such large insect decline as they are facing in the Northern Hemisphere. So the Cape Floristic region is the smallest floral kingdom, and it is threatened by land transformation for agriculture, it is threatened by land transformation for agriculture, land development, as well as alien plant invasions. And climate modeling exercises have demonstrated that the CFR is likely sensitive to climate change, and the fynbos biome in particular will lose large of its natural vegetation near its northern limits, especially those along the west coast and the Cederberg Mountains. So given these concerns and the loss of biodiversity, conservation strategies were put in place in the Cederberg. However, these focused on medium and large animals. So small animals such as arthropods have been neglected, and a prime example includes ants, which are important seed dispersers, especially in the fynbos region. So I'm just going to tell you more about the seed dispersal. So ants benefit on seed dispersal by eating the juicy, lipid nutrient-rich part of the seed. So that's the white liquid. So the ants carry the seed to the nest where they feed the whole colony. And then once they are done eating the juicy part of the seed, they then discard it. So when they discard it, they either leave it in the nest or discard it outside of the nest. And then this benefits the seed in that if the seed is kept inside the nest, it is protected from factors such as predation as well as fires. And the seed that is discarded outside is also benefits because it has been transported to more rich nutrient soils. So the objectives of my study were to assess changes in ant assemblages over 20 years along an altitudinal gradient, as well as assess which environmental factors best describe these changes in ant assemblages. So the Cederberg Wilderness this study in the Cederberg Wilderness area was initiated by Prof. Stephen Chown in 2002, and sampling took place from 2002 to 2012, and we have a beautiful reference collection for those years. So for my study, sampling recommenced in 2022, which was exactly 20 years since the study had started. So we sample the exact sites that were sampled previously, which is 20 years ago, and then we take note of changes that have occurred over time. There's been papers that have been posted, that have been published, such as a study published on ants by Borders, and there's more, such as studies published on beetles by Gauns. So the transects in the Cederberg represents 3 vegetation types, and the first vegetation type is the strand-fed succulent karoo, which is characterized by broad-leaved and small-leaved succulent shrubs. The second one being the lowland succulent karoo, which is characterized by succulent shrubs as well as annual grasses. And then the third one is the mountain fynbos, which is characterized by restiodes, proteas, and erica, and this encompasses most of the transect of the Cederberg mountain. So the transect starts at Lambert's Bay, which is at the beach, picking up at 1,900 meters above sea level at Snoekop, and down to the eastern slope at the village in Wuppertal. So a total of 17 sites, which were at 200-meter intervals, were collected. And each site was replicated 4 times. The replicates were at least 200 meters apart, and each replicate consisted of 10 pitfall traps. And this made a total of 680 pitfall traps collected per season. The pitfall traps were then half filled with 99% propylene glycol, and these were left in the field for 5 days. So in the lab, we then washed the samples and stored them in 96% ethanol, and then ants were mounted and identified 2 species. So I then also re-identified the historical collection, matching the species with the newly collected species. And I did this using morphology, which is sitting in the lab for 24 hours and not going home. So this is where barcoding is useful, and Dr. Charlene will talk more about that. So I also collected environmental factors such as surface coverage. So here I estimated factors such as background, rock, litter, and live vegetation using a sampling grid, as you can see on the left. I also used, we also did plant height, where we counted the number of species touching the measuring pole at different segments. And I also collected soil to get some soil properties of the soil around the cedar bag. I will also used eye pattern sensors to measure soil temperature. These were planted at 1 to 2 centimeters under the soil, and this was started in 2002, and this is still ongoing. So we do have temperature data over 2 decades now. So I did just an NMDS and RDA on R. So in South Africa, we have 732 valid ant species. And just in the cedar bag alone, 115 species were identified. And this is just 2 years of my PhD. So meaning that if we identify more years, there might be more ant species. So the most abundant species were Monomorium freedi. This is also included in our species cards. It was Anoplolepis custodians and Anoplolepis stengroveri were also very abundant. So there was a significant decrease in ant abundance, from March 2002 to March 2022, but in October 2002 to 2022, there wasn't really much. But what was important to note here was that as you go up the mountain, the abundance decreases in both the seasons. So as you can see at 1,900 meters, this is the peak, that is the western slope, and this is the eastern slope. So here you can see that it always has the least abundance in both the graphs. Same as species richness. However, there wasn't so much here, but what was important to note here was the significant decrease in species richness from the years to this current years. I also ran an NMDS to see the community an assemblage of ants, and it showed that there was a significant difference in ant assemblages between the previous years and the current years. This might not be as visible in the NMDS, but as we do altitude, like an NMDS per altitude, then the differences become more clearer, which is what I'm doing for my PhD. I also ran an RDA to see which environmental factors best describe the changes in ant assemblages. So all these factors that I've listed here did have a significant impact. However, altitude, pH, and mean temperature explained most of the changes in ant communities. So the changes in ant assemblages between the years and the altitudinal gradient were the result of change in temperature, but this was altitude specific. So some temperatures were not really that much affected, and some were more affected. And also it was the result of changes in vegetation structure. So we noted an overall significant change in background. So background significantly decreased, and then there was a significant increase in litter across the altitude. But with vegetation, which is live vegetation, there was decrease and increase at different altitude. So altitudinal studies play a role as proxy for climate change impact. And long-term studies are crucial for documenting changing biodiversity. And this study in the cedar bag is one of the long-term studies in Africa assessing the impacts of climate change over time. So I'll give Dr. Charlene.
Thanks for that nice introduction, Busi. So I'm going to tell you a bit about, or before I go further, Busi will actually, she won a prize to represent the mountain community of South Africa, and she will represent this work at an international conference in Austria in September. So that is really fantastic. So as Busi did a lovely introduction of the area and of the Cederberg and the diversity, I want to tell you a bit about the usefulness of molecular analysis. And as I tell you all, we're not molecular biologists, but we really, really appreciate it as a tool. So as Busi said, we're working in the Cederberg, but there's actually 3 transects that were initiated in 2002 by the Center for Invasion Biology. And one was in the Soutpansberg, one was in Sani Pass, going up to Lesotho, and then we have the Cederberg transect. So these 3 transects were started at the same time in 2002, and they're still ongoing. Sani Pass has been going for almost 20 years. We had a bit of a gap, and Soutpansberg also had a bit of a gap. But regardless, we all have data for about 2 decades. So now the challenge is to compare ants between these 3 regions. We use exactly the same methods. We use pitfall traps as replicates, and we put them out for the same amount of time, and we all meet on a regular basis to compare data. But now there's quite a challenge to compare species over time. And this is a slide from Busi. She mentioned, what she didn't mention is that she worked through thousands of samples, so we don't have a problem in the number of specimens we have. Although 80% of those would be dominated by one species, Anoplolepis, which are the great seed dispersers. But just to give you an idea, Busi spent 2 years of her life sorting, a full-time technician, so you can just see the costs are actually astonishing when you get to getting a result of what is the name of the species. And having a name is really crucial for us for comparing regions, but also for conservation efforts. So to identify an endemic species or very unique species, taxonomy is a very long process. But I'm a taxonomist, so I'm not going to say leave the taxonomy. But we really like to combine molecular and taxonomic methods. So how do we actually do this? So we use molecular analysis to firstly verify taxonomic identification. This is an example of ants from the Americas, North and South America, and just to give you an idea of what's into all the delicacies involved in ant taxonomy, looking at different parts and hairs, and Busi is a trained ant taxonomist, learning from the best at Izika Museum. But as you can imagine, it takes a very long time to actually get into this and identify. But when you compare it with some molecular methods, and we've tried a lot of barcoding, we have a big barcoding library, but CO1 just doesn't work for ants. And Anya can probably also talk hours about that. They're difficult. Most insects have got a hard exoskeleton. Sometimes the color actually influences the extraction for some reason. So for ants, more and more they're using UCEs. I just want to get this right because, as I said. So UCEs, as you might know, it's a region of the genome that is shared between the distant taxa, and that is then shows them that it's very useful for tracing the evolutionary history. And so that's been used a bit more to look at where species overlap and how the clades then differ. So UCEs have been more and more used for ants. So this is where we want to explore that a bit more. And in Busi's master's thesis, she looked at only Anoplolepis. And this is specifically, if you look at the red dots, Anoplolepis species 3, that's also something that we need to highlight. Most of these species are undescribed, so we have to give them names. But even if they have amorphous species, they overlap here. And how do we actually know if they are real species or not? And this is where UCEs come in very importantly. And also going back to, if you think of, if you look at this map and this, so these species are overlapping in our 3 transects. So it will, if it took Busi 2 years to go through these samples, it will take 3 times that to go through 1 season. So we are trying to find ways to sort of fast track so we can make these comparisons and say which species are unique in all of these transects to aid in our analysis. Another way that we use that is to understand the historical processes, and this is where UCEs also come in and the genomic work come in very handy. And there's a great example here from our colleague, the curator of the Hymenoptera section at Iziko Museum. So they're also on board with the 1KSA project. Nokutula Mbanyana Ntleko and Simon van Noort. Nokutula is an ant taxonomist. And she did this really lovely paper on Ossimyrmex. So Ossimyrmex is an ant genus that's usually in more arid regions. And she used UCEs to look at their divergence patterns, and she found that they probably originated in the southwestern region of southern Africa. And just to show that all these climate changes, all these long-term climate changes, resulted in their diversification. So this was a really useful example to see how certain genera radiated and how we can actually focus our taxonomic expertise on a certain genus. So we can't be an expert on every single genus or family of invertebrates, so this really helps to focus funding especially. And this is Ossimyrmex barbica that was from the Cederberg, and this was sequenced, thank you to the Diplomics team. This is Ossimyrmex barbica. So this is just from ant maps. It's an amazing database that we use to just look at the distribution of this specific species in southern Africa. You can see it's quite widely distributed. So this is one of the species cards. There's 3 arthropods on the website, and I'm happy to say they're all ants, okay? The next one, Tetramorium, and you can see it's quite widely distributed in Africa. So some of these species have a worldwide distribution, and I'll get to that now. But this is also quite a lovely species. And then Monomorium fridae, more restrictive distribution. And you'll see from the metadata, we sometimes don't even know the biology of some of these species. We know if they're great ant dispersers or what they feed on, but this is an example of we don't know much about the biology even, which is quite remarkable. And then lastly, this goes a bit with the first aim, but it's really important to detect invasive species. Invasive ants can have really detrimental effects on our local ecosystems. I don't know if you all know about the Argentine ants. They are pretty much everywhere in South Africa, but they are bad seed dispersers. Brucey told you how good our seed dispersers are. Argentine ants would just grab the seed and they would just drop it. They would eat it and they're very bad seed dispersers. Luckily, yet they're not in pristine areas, so they're just around disturbed areas. Those are the ones in the Western Cape, at least, you'll find in your home. So detecting some of these invasive species are very important before they actually get into the country. And a great example is Anoplolepis gracilepis. They are worldwide very invasive. They're on the top 100 invasive species in the world. When they invade an area, they're very abundant in Australia and around the islands, especially on islands. What they do is they do what ants do, and they actually farm with aphids. So they look after aphids and protect them. The aphids basically have they have honeydew that they produce, so because they plant suckers, and this creates mold and just huge agricultural impacts. Not only that, they will attack anything. They attack crabs and geckos and vertebrates. On Christmas Island, they actually attack the coconut crabs, which has led to an ecosystem meltdown where the decomposition is actually impacted. So even a little ant can have massive devastation. There was a record, that tiny record there in South Africa, and Brucey, having a background in the museum, she's like, no, I've never seen this species. So we actually did a whole chapter with Brucey where we sampled everywhere along harbours, because this isn't actually an iNaturalist record. It's not published. So there's no published records of the species. It was an iNat record, and we went to verify it or not. And we say it's definitely absent. We sampled everywhere. We looked at all the museum records. So it's not in South Africa, luckily. But this is an example of a species we should flag at, for example, harbors and airports, and go if you see this. And it's very easy to confuse this with another species. They look very similar to some Solenopsis species. So someone would take a photo and say it's here and create havoc, or someone could miss it. So here is where having a nice molecular basis and library could be really useful to prevent, and this goes for all invasive species in general. Just to wrap up, in terms of our 3 transects, we focus on ants, but we also have a student working on the beetles, on the beetle body sizes, going up in altitude. We're very interested in the spiders. I work on Calimbula, and really all the taxa we send to taxonomic experts to verify. But again, the time consumed, again, we just work on ants now. Before we started our relationship with Diplomics, we started looking at how we can take a pitfall trap. This is a pitfall trap which could have hundreds of invertebrates in, and see can we maybe do meta-barcoding. But because we didn't have a library, they still have to do barcode by barcode. But I just want to show you the results from this. Is from one site's pitfalls. They found 1,474 unique bins. That means 70% of the species in the traps in that site has never been barcoded even. So that is, for me, overwhelming, but it's also encouraging to go, let's work on developing rapid methods, because we can lose species faster than they are described, because most or half of the species are undescribed. And I think invertebrates are a great proxy. They're small. Permitting issues, not so much, so we're lucky with that. So we want to try and just fast track this, and we're quite excited to work with Diplomics. And again, thank you. We were hosted for a workshop, which was amazing. I learned terms like qubit, and I throw that around now. But thank you so much to Anya. Thanks to everyone on the team. This is Ati, has been working really closely with you guys as well, because she's battling to get data out of tiny little fig wasps, and they're museum specimens. So that's another challenge for us, is degraded material. We can't go back 20 years and collect an extinct species. So that's also something that I think the nanopore technology can help a lot. So we're very excited, and everyone was having a great few days. So thank you very much to the team. We have a lot of volunteers over the 20 years that helped us. Thanks to Diplomics for helping us with this video. You can go and look at the video about our project. And thank you for your attention and having us today. Thank you.